coming up. I'm thinking about the incredible science that has come out of Israel that has brought so much healing to our people and to the world. Tonight should be a great night of learning, and we're delighted to have you with us.
Thanks, Rabbi.
Okay, I would like to introduce Jay.
Thank you.
Who's going to introduce our speaker.
Okay, thank you, Rabbi. It's my great honor to introduce our speaker for this evening, Dr. Gary Grad. Gary did his medical oncology training at the University of Chicago before joining and becoming a partner at Northwest Oncology and Hematology in the Northwest suburbs. There he has had an active clinical practice for over 26 years. In addition to his clinical practice, Gary is consulted for several pharmaceutical and biotechnology companies, as well as local, regional, and national hospital and healthcare networks. Gary served on the Chicago ICRF board for 5 years and the International ICRF board for the last 2 years. So Dr. Grad, thank you for your service, and take it away.
Thanks, Jay. I really appreciate you for helping us come in and do this part of the Israeli Innovation Series, and appreciate Anshe Emet, not just for this series, but really being a role model in the community for what a conservative synagogue should be. Growing up at Northwest Suburban, and then most of my adult life has been at Beth Shalom, where I served on the board and executive board for many years. We would always look to Anshe Emet as sort of the example of how it should be done, and we're really grateful for that in the community. I got to know Rabbi Siegel a little bit, standing in lines outside of Camp Ramah, waiting to go in on parents' weekends. And those of you who've been at Camp Ramah know exactly what I mean. And Chazan Mizrahi always holds a special place in my heart. My father, Zichronon Livracha, was a huge fan of Chazanut and loved Chazan Mizrahi. And back in 1998, at a memorial service in my father's memory, he was gracious enough to come and actually perform at Northwest Suburban for that event. And he'll always hold a special place in my heart personally because of my dad's connection to Chazanut. So with that, let me see if I can get my screen shared here. And here we go. Share. Oops, that's not what I wanted to share. This never works the way it's supposed to. Can you guys see my screen?
Yes, we can see your screen.
Okay, you can see the slides. Okay, terrific. Thank you. So we're going to talk a little bit about Israeli cancer research, and I entitled this A Light Unto the World, partly because of Hanukkah, partly because I really think that's what Israeli research is all about. And our motto is that the answer to cancer is research. And I think the best research in the world, for a number of reasons, which hopefully we'll explore tonight, is coming out of Israel. And let's see if I can advance my own slides here. There we go. So I thought I would start on a personal note, if you'll indulge me for a little bit, just to give a little bit of my background. You've heard about my medical background, and I'll kind of explain how I came to be passionate about Israeli cancer research and ultimately ICRF, as opposed to what's going on with cancer research elsewhere in the world. I mean, I've spent most of my professional career involved in cancer care and cancer research here in America. So why Israeli cancer research? My Jewish communal endeavors revolved around JUF, going back to young leadership with a lot of people who grew up in Anshe Emet, actually, back in Hyde Park for many years. And subsequently, I'm sorry, I have all kinds of technical problems here today. Subsequently, I got involved in APAC and some other endeavors. I was fortunate enough to be a Wexner Heritage Foundation participant back about 20 years ago. And through that process, met a rabbi who was one of our directors and educators, Rabbi Laufer. And one of the things he said really struck a chord for me. And he said, if you want Torah to become alive, you have to take God out of the story and put the prism of your own life narrative in the story. And I was always fascinated by that. And because of that, as I went through starting to learn rather late in life, there were some passages that kind of resonated with me guiding my Jewish communal work. And one is a phrase, L'or Goyim, which comes up in Torah, light unto the nations. One can do a whole dissertation on how that meaning has been interpreted, whether it's been liked, whether it's been disliked by different groups with different ideologies, political, religious views. That's for another day. But for me, it really means what Rabbi spoke about earlier, and that is bringing a light into the darkness in the world. And not just in your personal life, not just in your own home, not just in your community, not just in your country, the nations of the world, but the entire universe. But as a cancer doctor, to me, that means bringing light into the lives of those in darkness, which is driven by a diagnosis of cancer and for their families. Another Torah passage that resonated with me was Balaam as a representative of those who are set on cursing us. And yet when they look down, could only say, Matovu Ahalecha Yaakov, that even when they want to curse us, what's coming out, what's emanating out is only something that they can look down and ultimately praise and say Matovu. And then finally, I have also kind of felt the legacy of Ruvain, Gad, and half Manasseh, the 2.5 tribes who go to Moses and say, we want to stay on this side of the Jordan. So as a Jew, as someone who is a staunch Zionist, who loves Israel, I've had to answer for myself many times over the years, why am I here? If I want to, if I believe that strongly, why am I not there? And so I stay here for a number of reasons. And I do it with the legacy of the 2.5 tribes, understanding that that's okay and I can stay connected to those in the land, but only to the degree that I'm part of the vanguard of defense for my brothers and sisters in the land. And so interpreting what that means through the prism of my own life is how can I use my professional skills as a cancer doctor and have that resonate for me. And there are certainly other things I've done. There's lots of ways to do that. In the old days, it was grabbing a sword and fighting. Now it's fighting on a geopolitical scale. It's giving support financially and through working for organizations that support our brothers and sisters in the land. But again, if I look at it through the lens of the prism of my narrative, it's that of a healer and a cancer doctor, and how can I use that to help Israel in that setting and still feel connected? And then, of course, like most things happen in the Jewish community, one evening at a Shiva, I happened to be sitting next to the professional who was working in Allie's role as the executive director of the Chicago office of ICRF. And she started telling me a little bit about ICRF, and my initial reaction was, well, my dance card's full, which is sort of my knee-jerk response sometimes. And as I sat and thought about it, it started resonating with me that this was really where I was meant to be, that this is the culmination of my Jewish communal work that can meet all of those 3 phrases which have always resonated with me since that early learning through Wexter and Rabbi Laufer. We recently had our international board meeting, and Rob Dennison, who's our outgoing board president, in his speech basically summed it up as well as anybody. He said, I love Israel and I hate cancer. And that pretty much brings me to why I've gotten involved the way I have, exploring Israeli cancer research and innovation, and ICRF. So those are the 2 questions I want to spend some time on tonight. What's unique about Israeli cancer research compared to anywhere else in the world? And then I'll spend a little bit of time about why ICRF. So that little picture on the screen is meant to be a nutshell. So this is my version of sort of cancer research in a nutshell and the way it's gone on for decades. And the x-axis is time and the y-axis is time, and the lines are quite long. And the way cancer research has traditionally worked is at the very bottom arrow, we have something called bench research or basic research. At the very top, when something finally comes to the clinic, we call that bedside. So the goal on that y-axis is getting from bench to bedside. Basic research or bench research are concepts at the very basic level. They might be biologic concepts, chemical concepts, molecular concepts. They may or may not have anything at all to do with cancer research. They're basically understanding the basic biology or chemical reactions in a biological process. Eventually, those concepts are looked at and said, hey, there's something here that we might be able to apply towards cancer, or there's something here that we might be able to target as an agent in cancer. And eventually we go through identifying those concepts and bring them to preclinical models. In the old days, that was animal models and cell lines that are cancer cells cultivated in a petri dish or injected into animals to grow tumors in animals. Nowadays, there's some fascinating stuff going on with computer-based models and organoids where you can build these 3D dimensional copies of genes and proteins. It's really pretty fascinating stuff in terms of the computer engineering. And then ultimately, things are identified that can be brought for clinical trials. Agents, drugs, targeted agents, immune therapies, and those are tested initially after they're done in animal studies, are brought to people. And the phase 1 trials are just looking at the safety of these drugs and different dose levels of the drugs, not even efficacy. And from there, you see where the drugs work and you take it through the process of eventually getting a drug to market. The problem is that concept can take years and years and years and is extremely inefficient. So I've incorporated the help of one of our early funded scientists who went on to win a Nobel Prize, Dr. Chickanover. His early research down at that bench level at the bottom arrow had to do with uncovering a pathway that eventually led to the development of a drug called Velcade, which is used every day now to treat multiple myeloma as a standard of care and has really impacted the lives of a myriad of patients. And I'll let him explain what the time frame is.
You don't have his, yeah, you don't have his voice thing on. When you share your screen, you have to make sure the little button for sharing sound is also pressed.
Okay, let me try that again. Where's the sharing sound button? Let's see. I have a volume button. Share sound. I'm sorry, guys. Does anybody know where that button is to share sound?
You may have to go into New Share and then select your screen again, and there should be 2 little options at the bottom of the screen that say Share Sound, and then you have to.
I've got it. Yes, thank you. I'm sorry. Let's try that again then. And let's see if this works this time. I'm so sorry, guys. I'm going to reset the sound here and see if it'll play.
Very gradual. You know, if we are looking at the timeline, I joined Avram in 1976 when I was discharged from my service in the Israeli military as a combat physician. I finished my PhD in 81. Avram offered me a position, and the first drug was approved by the FDA, the proteasome inhibitor, just before we got the Nobel Prize in 2004. So we are talking 28 years.
All right, can everyone still hear me?
Yes. Yep, we hear you fine, Gary.
Okay, great. And you were able to hear the video, yes?
Absolutely, yes. You got it.
Okay, terrific. So yeah, so that timeline, that y-axis, you can hear from Dr. Chickanover, from that bench research up until a drug was actually approved, took 28 years. And it just highlights a little bit about the expectations in cancer research, that we plant seeds now that might not yield treatments for a long time. However, I think because of what's going on really being led in Israel, but a lot of other places, I think we're going to be able to accelerate the efficiency of that timeline. And I'll talk a little bit about that in a second. So why is Israeli cancer research different than research that goes on at University of Chicago, Northwestern, Johns Hopkins, the National Cancer Institute, Harvard? And why do I have this slide up? This slide is Ronit Sachi-Feonaro. And Ronit is the head of physiology at Tel Aviv University. She runs a very large lab there. She is one of the most cited cancer researchers in Israel. And she is a ICRF funded researcher, so I've had the chance to get to know her a little bit. And I actually sat down with her. Her research focuses on what we call nanotechnology and applying that to cancer metastases to understand how cancer cells metastasize to the brain and elsewhere. she was actually able to adapt a lot of her platform from her lab very rapidly to help with the Israeli efforts on COVID research and some of the nanotechnology being used in COVID research. But I asked Ronit once when she was in Chicago and we were having a cup of coffee, what is different about Israeli cancer research? She did most of her training in the States, but did her postgraduate training at some of the top institutions, top institutions in the state. And then she went back to Israel and heads the lab at Tel Aviv University. And these are some of the things Ronit came up with for some specific reasons for why she thinks things are better in Israel or more conducive to innovation in Israel. Number one, there's an established academic hierarchy at all universities, and it usually goes from student to grad student to postdoc to junior faculty to professor and eventually the lab or research director. In the States, Ronit found that there was sort of a strict adherence to this. And though there were meetings where people can share ideas, you really never bypassed the person above you in the hierarchy. That's not what the culture was. And in Israel, it's a very different culture where if you have a good idea, you're able to bring that sometimes to the highest levels, as opposed to having to go through these multiple levels where sometimes good ideas get filtered out before they should. And that was an important aspect of Israeli research that she certainly runs her lab with that philosophy, but she really said most of the labs run with that philosophy at Israel, that's a unique cultural issue there. Number two, she also said that there was more of an ease and even an encouragement sometimes to shift to tangential ideas that arise as you're studying a particular idea. So if you get a grant to study concept X in the United States, you pretty much see that grant through, and you're rather discouraged from going off and branching off if you find other concepts that might arise that are interesting. And in Israel, that's not necessarily the case. If you find something serendipitously that might be more intriguing or more relevant, you're actually encouraged to go and pursue that. Number three, just the basic economic efficiency. Israelis can do more with less. There's more of an appreciation because the funding is a little bit less, quite a bit less than it might be in the States, that they have to do more with less. They have to be more economic with their labs, with their materials, with how they use or waste things. And they are much more efficient economically with the funding that they get. And finally, and Ronit was a little uncomfortable mentioning this because it's a little bit elitist, she talked about this intellectual density factor. And you know, here you can walk into a coffee house and you might find some very bright people from a whole number of different professions, and have some interesting conversations. But it's not the same as in Israel where you can walk in a park or on a beach or in a restaurant or in a coffee house or before or after, if the rabbi closes his ears, sometimes even during minyan, and meet people who not only have incredible levels of intellect, but also are pursuing things that are relevant to your field, either directly or tangentially. And you have these conversations about politics and about what you're doing that lead to new ideas. And the density of that intellect is higher, and it increases the chances of those conversations happening. This is an article taken from Israel 21C. Israel 21C is, for those of you not familiar with it, is an online magazine that really highlights Israel in the 21st century. And if you haven't checked it out, it's certainly worth checking out. We have a Chicago connection to this. The new executive director is Brian Abrahams, who many of us know. Brian was my fraternity brother and a friend for many years. He was also the Chicago director of APAC for many years and has been involved in a number of Jewish community activities. So Israel 21C talks about bioconvergence as the next big thing from Israel. And their subtitle is "Drug Development Will Increasingly Be Driven by a Sophisticated Overlap of Biology, Computer Science, Mathematics, Engineering, and Nanoscience." And again, all of this stuff is going on at multiple places and in different silos here in the United States. What's unique to Israel are those factors I mentioned before: the culture for the type of collaboration that's there, the intellectual density that's there. And the hope is that we can accelerate the efficiency of this bench to bedside timeline, as well as observational research using machine learning and computer science and artificial intelligence. And so if you look at that curve again on the left, we're hoping that we can dramatically shrink that timeline. And I guess a way to sum it up is if I were to sit here in the suburbs of Chicago and tell someone else, a biologist, a computer scientist, a mathematician, 2 engineers, a nanoscientist, and a rabbi were having coffee one morning in Tel Aviv, the comment would be, "Nu, what's the punchline?" If you say something like this in the streets of Jerusalem or Haifa or Tel Aviv, someone's going to say, "Nu, what did they come up with? What's the new idea?" And I think that really kind of highlights that intellectual density that Ronit was a little uncomfortable talking about. This comes from an online journal called No Camels Team. No Camels is a online journal. It's meant for global consumption. And what it is it's about Israeli innovation and technology. And they break it down into different genres of innovation and technology. This is taken from their healthcare. And this was a top 5 recent advances in Israeli cancer research and treatment. And I thought it's an interesting list to go through. We can go through some of these top 5. This goes back to February. The list is probably a little different now. Some of these researchers are actually ICRF funded. Not all of them are. So we have relationships with many of them. And I thought I would just go through them. So the first one is from Professor Yuval Shaked from Technion. And it's about understanding resistance to cancer treatment. Now, a lot of people are studying resistance to cancer treatment, usually from the standpoint of genetic mutations that evolve once someone is on treatment, that make the treatment no longer work, make the cancer cells resistant, or you have clonal selection where you treat the sensitive cells and then the cells that are inherently resistant are the ones that proliferate. Other people look at mutations that arise irrespective of the treatment that just lead to resistance of a treatment. What Yuval is looking at is what is the host environment like that might lead to an understanding of resistance? And certainly with all the immunotherapy and immunobiology going on right now in cancer research, that's a very important process to look at because we're understanding that it's the host environment sometimes as much as the cancer cells and their own genomics and genetics that affect sensitivity or resistance to a treatment. So some pretty interesting research coming out of there that was rated by No Camels as one of a top 5. The second one is from a company called Alpha Tau Medical, looking at a new type of radiation therapy called diffusing alpha emitter radiation. And again, there's a lot of thought that that's going to be a big breakthrough in how we deliver radiation therapy to cancer cells. The next one involves Malka Cohen-Armon and Talia Golan from Tel Aviv University Sheba Medical Center. The first one is this compound PJ34, which is a targeted therapy for pancreatic cancer, certainly a cancer that has deep meaning for a lot of Ashkenazi Jews where the incidence is higher because of things like BRCA. We'll talk about that in the next article. And any new breakthrough in pancreatic cancer is always embraced with great joy because it's such a difficult cancer to treat. Talia Golan is mentioned a second time in this top 5, and this is for her previous pancreatic cancer research. She was the lead researcher in an article published in the New England Journal of Medicine in 2019. This was also presented as a plenary session, meaning one of our key practice-changing sessions at the American Society of Clinical Oncology meeting, which is held in Chicago every year. This year, unfortunately, was held on Zoom, but the last one that was held in person back in 2018, this paper was presented by a Chicagoan, actually. Dr. Hetty Kindler from University of Chicago presented the article. She was also a co-author on this paper. And this looks at pancreatic cancer patients who harbor what's called a germline BRCA mutation, meaning that they have the hereditary BRCA mutation. Pancreatic cancer is increased in incidence in those patients. And there's a class of drugs that we've learned about in other disease states like ovarian cancer, prostate cancer, breast cancer, that if you harbor these mutations, these drugs called PARP inhibitors can have some efficacy. And what Dr. Golan and Dr. Kindler presented and is now considered standard of care is that for patients with advanced stage pancreas cancer, if you harbor a germline BRCA mutation, after you receive chemotherapy, if you've been stable or improved on chemotherapy, then you go on these PARP inhibitors, and it dramatically improves the progression-free survival and the overall survival of these patients to go on these PARP inhibitor drugs in that setting. So it's been really practice-changing and life-changing for patients with BRCA mutations in pancreatic cancer. And again, that's an innovation that came out of Israel. And then finally, the fifth one on the list is Dr. Tzvi Livne and Tamar Paz-Elitzur from Weizmann. And they came up with a DNA repair biomarker score that can be looked at to predict lung cancer risk amongst higher risk patients, who's at risk to get lung cancer. So these are all articles that this Journal of Innovation and Technology highlighted in their healthcare section as coming out of Israel in 2020. So it's just a smattering of some of the amazing work that culture and innovative environment I spoke about is producing. And as you can see from Talia Golan, practice-changing and life-changing innovation that's coming to the bedside very quickly. So I want to shift gears a little bit and talk about why ICRF. And the two are related. ICRF fundamentally believes in the unique intellectual, creative, collaborative, and innovative culture of Israel to be optimally positioned to change the world through innovation and disruptive cancer research, thereby improve cancer care for people worldwide. Therefore, ICRF fundamentally believes in the value of funding Israeli scientists in Israel so they can have adequate resources to remain and continue their work at home and not need to seek appointments in Europe or the United States. Funding from ICRF is institution agnostic. It does not depend or exclude any institution. It's based on the research and the researchers. We have a world-class scientific review panel. So unlike giving money just to a particular cancer organization or institution, our research is funded by a world-class scientific review panel that gets together annually and only funds grants that meet the highest level of scientific rigor and criteria and scoring. The funding that we give is to the researcher, not the institution, and that's different than a lot of grants given by other grant sources, both here and in the United and in Israel, where a lot of the funding goes for the research and a lot has to go to the institution. All of our funding goes to the researcher, not the institution. The grants are usually multi-year grants to allow projects to continue. And then ultimately, there's a lot of prestige that Israelis get with an ICRF grant, and they can leverage that prestige for additional grant funding down the line as their research progresses. And that's been a great source of additional funding, and many of the Israeli scientists continue to tell us how useful that is as their research progresses. I mentioned our scientific review panel. We have 40 to 60 world-class scientists that get together on their own time every year. Over the last 2 years, we've given out about 70 grants a year, $4 million annually. We've been able to continue to raise funding despite COVID. And where there's a lot of cancer research support drop worldwide, we have been able to continue to support all of our grants and ongoing Israeli cancer research. We know that cancer does not stop for pandemics. This is just a snapshot of what's going on now. Over the years, we've given out about $72 million in the form of about 2,500 grants. The active grants we have right now on the left column, you can see the areas of research that we're funding, and some of these are bench research and some are more bedside research. In the middle column, we have the disease states that are being studied by these researchers. And finally, on the right column, alphabetically, we have the institutions that are currently housing those researchers who are receiving those grants at the current time. Those are the active grants. I'll close with, I have a summary at the end, but I have some of our researchers. Hopefully the volume will work. Someone let me know if it doesn't. And I have some of our researchers, just some quick clips, just saying thank you.
I have a very unique history with the ICRF. And let me first say this summarizing sentence before I go into the details. I owe the ICRF a debt that I will never be able to repay.
My research deals with the p53 tumor suppressor. Thank you, ICRF, for supporting my research.
My research focuses on elucidating the interactions between brain metastases and the microenvironment. Thank you, ICRF, for supporting my work.
I would like to take this opportunity and thank the ICRF for actually supporting me right at the beginning of my career.
My research is about NK cell killing tumor cells. Thank you, ICRF, for supporting my work.
So just to conclude, Israeli cancer research with the help and support of ICRF and many, many others is changing the world. I hope I've been able to show you a little bit of that tonight. Like the Chanukiot, which will adorn our windows next week, Israeli cancer research brings a shining, healing, and inspiring light of hope to the darkest places for those with cancer and their loved ones. As Hanukkah celebrates the rededication of the sacred, let us all choose at this time of year to rededicate ourselves to help and support the miraculous discovery and innovation of ongoing Israeli cancer research. The answer to cancer is research. Thank you.
Thank you, Dr. Grad. There are a couple of questions actually in the chat that I wonder if we could take a moment with. Alice asked a question here. She said, isn't there research by Professor Malov at the Technion going on?
So I certainly don't have a list of all of the comprehensive research going on. We can certainly look into that and get back to you with more details on it. There is tons of research going on that I am unaware of, both here and in Israel. If there's any specifics though, we're certainly happy to have a follow-up conversation. We can let you know if we're involved. We can certainly reach out if it's someone we're funding, but even if not, we can certainly touch base and let you know about the details of their research and the potential applicability of it.
Thank you, Doctor. There's another question here from Cheryl, and the answer might be the same. But Cheryl was wondering, are there any new developments in BRCA2 mutation to avoid double mastectomies?
So it's a great question. And the truth is, right now, that patients who carry a BRCA2 have an increased risk of breast cancer. And if they've had breast cancer, they have an increased risk of another breast cancer. They also have risk of ovarian cancer. They also have risk of pancreatic cancer and prostate cancer. So there's a number of ways to reduce that risk, and it's all about risk and benefit. So there are some therapies out there that can lower the risk. The only way to really get the risk down near zero, and it's not completely zero, is with double mastectomy. The question is, are there any innovative treatments that can prevent the risk completely medically without having to do double mastectomy? So are there women with BRCA2 who choose to not have double mastectomy and to take drugs to lower the risk of cancer, such as aromatase inhibitors or tamoxifen in premenopausal patients, and do those lower the risk? They do some, but often not low enough. You're still dealing with a high risk. And so women have to decide on themselves, what risk are they willing to endure? These newer drugs like PARP inhibitors are not really being looked at in the preventive setting. Most of those are being looked at in the treatment setting. So even if a woman has a breast cancer and has a BRCA2 and has what we call breast conserving therapy, meaning a lumpectomy and radiation, unfortunately, there's still a high risk of recurrence in that other breast. And some of that decision process is personal. Some women say, I understand the risk, I don't want a double mastectomy. Some of it might be age related. If you're 85 years old or 90 years old, then the risk, because your expected life expectancy is less to start with, the time frame of which you might develop another cancer might be less, and you might decide not to do that. But right now we don't have a magic way of eliminating the risk of breast cancer or these other cancers in high-risk patients without surgery, which is still the best risk reduction, and even that's not 100%. It's close, but it's not 100%. When they do a mastectomy, they can never get every single breast cell out of there. They have to leave enough tissue still, where there's always the risk of some rest of breast cells there. Fortunately, the risk is exceedingly low. So the closest to 100% risk reduction for breast cancer in BRCA patients is surgery. Same thing with ovarian cancer. So right now we're still looking. We hope to find something magic that can alter the BRCA2 gene effects, but we're still a ways away from that. But is it being looked at? Yes, and I think a lot's going to be done as we start unfolding and understanding the three-dimensional protein structure from these gene mutations, or can we revert these gene mutations into more normal gene-like phenotypes? And there's a lot of work being done with that. We're actually starting to understand now women who have advanced breast cancer, for instance, who are on a PARP inhibitor, sometimes get a second mutation in the BRCA that restores it back to original BRCA function and makes it resistant to the PARP inhibitor. But if we can understand how that gene works, can we then induce that and prevent women from getting the cancers associated with BRCA and induce more of a BRCA normal phenotype. So there is a lot of research going on, but I think on that timeline that I showed you, we're still fairly down low on accomplishing that or bringing that to the bedside just yet.
There's another question from Amy. Is the research in Israel more heavily focused on certain cancers, given the Ashkenazi connections to some of those?
It's a complicated, so the easy answer is yes, only because of the prevalence of that there. They have more BRCA cases because of the population density of Ashkenazi Jews compared to a place like the University of Nebraska, if you will. Now, plenty of non-Jews get BRCA mutations, but it is higher in Ashkenazi Jews. And so just by the nature of the demographics of the population, it's easier to study those because you have higher volumes of patients. Having said that, there's no limit to what's being done in Israel. You saw the slide where now we're getting into bioinformatics and computational and applying computer engineering to understand what are the genetic drivers. Do those gene mutations lead to RNA effects that are then expressed as proteins? And what's the interaction there? What's the interaction of the host? So again, at the bench level, everything's being studied and every disease state's being studied. But yes, BRCA gets a lot of attention because of the population density of BRCA in Israel.
Any other questions for Dr. Grad? Well, we thank you for enlightening us tonight, both in terms of some of the incredible cancer research that I think can give us all hope for the future, and the particular work that's happening in Israel with the support of ICRF. We're so grateful to you for sharing that, and grateful to Jay also for helping to bring ICRF here and to connect us with such an important organization, one certainly worthy of all of our attentions. Thank you so much.
Or there
Thank you,
was one
Rabbi.
more question in the chat room.
Oh, great, I missed it. Sorry, thank you. Right, Allison asked, what beyond BRCA should we worry about?
I guess the first question is, who is we? So there are a lot of gene mutations beyond BRCA that, oops, sorry, there we go. Beyond BRCA, which can increase hereditary risk for cancer. And this list is growing all the time. And it's driving a lot of the companies that do the hereditary testing crazy because they keep having to add to their panel. And there's things like CHEK2 and PALB2, which can lead to a whole Lynch syndrome. So what we're learning is that a high number of cancers are associated with hereditary risk genes that we never knew about. And I think the time will come within the next few years where anybody who has cancer will get tested for broad panel hereditary risk testing. Because if we use the guidelines now, it turns out that when we see patients with cancer, we're missing about 50% of people who actually have hereditary risk genes that don't meet the criteria for testing. They're not Ashkenazi Jews, they don't have family history, et cetera. So I think we're gonna learn a lot more, but there are gene syndromes that we're starting to learn about extensively. So really the time's gonna come where any cancer patient gets broad panel testing of multiple hereditary risk genes, not just BRCA based on a history, but it's gonna be across the board. The time may come where you get a blood test as part of your physical exam, looking not only for genes or methylation signals that tell you might be at risk for developing a cancer, but also whether you have hereditary risk for a cancer. And those are going to guide some of the pathways that develop then from payers and from hospital systems for how to screen people appropriately. So instead of these ubiquitous guidelines of colon cancer screening at a certain age or mammograms at a certain age, it'll be much more personalized screening based on these genomic risks. And I think that's probably the near future. And I would say that's probably next 2 to 5 years, we're going to start seeing that. So I think the whole scope of what we worry about will change. I think what you shouldn't worry about is not getting tested. I think anyone should get tested. Knowledge is power. There's really good protections against, right now at least, against discriminating against people based on their genetics. And you'd rather know so you can get into the screening, let your family members know if they're at higher risk. The other things to worry about is I wouldn't smoke. I would, wear sunblock. I would try to keep, weight in a good place. You still have to worry about heart disease and all the other niceties that we have in life to worry about. So I think you still have to take a common sense approach. And that would be my advice. So I wouldn't walk around worrying, but yes, there are a lot of other things besides BRCA which can lead to a whole host of cancers and cancer syndromes in families. So again, I think we're very near, probably within the next year, where every cancer patient is going to get screened with a broad panel hereditary test. And even healthy patients might be screened within the next 2 to 5 years as part of the routine.
Question, how effective is the genome in predicting cancer risk?
It's a great question. And this is where that bioconvergence that we talked about earlier in Israel. So what do you need to be able to do that? You need huge scale clinical data and outcomes data. And then you need huge scale genomic libraries, and you need to match those. And you need to apply machine learning and artificial intelligence analytics to that to start looking at the strength and validity of those findings. So part of the problem is that requires following people over time to see if they actually get cancer. So, you know, computer models for this stuff are great, but you still need to validate it. And then what do you do if you find it? And that's where we start getting into some of the bioethical things. So if you find someone's at increased risk to develop a cancer in 2 or 5 or 10 years, what do you do with that? You know, you make a mashuggi worrying about it. You could screen them, but you can't screen them every day, and you don't take major organs out that have life-threatening implications to go through a major surgery when you don't know for sure what the time frame is or if that person's going to get cancer. So it's a complicated question, but yes, genomics, I suspect, will be very good at predicting who is likely to get a cancer. They may not be as good as predicting who's not going to get a cancer, if that makes sense. In other words, there will be genomic signals or methylation signals that tell us that people are at certain risk based on things in their genome, that they're at risk for a cancer. We may not be able to tell, though, people who don't have those in their bloodstream if they're still at risk for a cancer for other reasons. We know genetics are not the only reason to develop cancer. Sometimes it's just age and background radiation and luck and a whole host of other things that can't be yet predicted.
Doctor, if someone were to take the BRCA test, would they be able to, would the results of the BRCA test tell the patient what cancers they might be susceptible to? If I don't think susceptible is the right word to use, but that's the only one I can say.
Yes, so if someone has a BRCA mutation, let's say you have a family member who's had cancer and is BRCA positive, and you take the test and you're BRCA positive, we know the majority of cancers that are associated with that mutation, and we actually have fairly good guidelines for recommendations for risk reduction of those cancers. And, you know, the hard part is, you know, again, if you're a young woman and you have a BRCA found incidentally, you've never had breast cancer, do you have a prophylactic mastectomy to reduce the risk? Do you go on medications? Do you go on a high-risk screening program? These are all options that have to be really thought through at an individual level between a clinician and a patient, going through the risks and benefits and the personal preference for that patient. But there are other cancers. In men, there's a risk of pancreas cancer if you carry a BRCA2, especially if you have a family history of pancreas cancer. It's a huge surgery to go in and take out the pancreas, and the risk is not as high as it is for breast and ovarian cancer. It might only be 8 or 10%. And so the question is, you know, how do you screen? So we do have screening protocols for trying to identify those at an earlier stage. And my guess is genomics will be part of those moving forward as we validate some of these findings we're seeing early on over the next 5 years. So yes, we can identify what cancers patients are at risk for a particular hereditary risk mutation, be it BRCA2 or PALB2 or CHEK2 or Lynch syndrome. And we do have pathways in place for most of those to try to catch these cancers earlier or have risk reduction techniques.
How effective is the treatment in Israel versus the United States? A number of Israelis that I know seem to have had amazing results from going to doctors in Israel who are Israelis. And I just seen there, from just a rumor point of view, it seems that Israel might be more effective in treating cancers than certain parts of the United States. Is that your opinion?
So you have to look at the data, and the data coming out of Israel, the United States, Germany, Asia. Cancer for cancer, really the data is all the same, and a lot of the trials actually being done are cooperative efforts that are international trials. You know, they may have certain clinical trials there. They're probably better at PR than a lot of American institutions might be with regards to their successes, although we've seen plenty of institutions in the United States get on television and certainly tout their successes. But in reality, I think the treatments are out there. We all know the same treatments outside of clinical trials, and a lot of the clinical trials are actually international cooperative trials too. So I would say that the treatments really are pretty equal in terms of their efficacy and pretty standardized. I mean, you're pretty much going to get the same treatment here as you would in Israel. One of the companies I work with now, we actually do a lot of work with Israel, and outside of an investigational setting in terms of standard treatments, you know, I see notes from Israel all the time, and it's the same standard treatments that you would expect at University of Chicago or at a community clinic, you know, here in the suburbs of Chicago or Evanston or anywhere else.
Did we get to Bruce's question?
No, let's take Bruce's question, and that will be the final question, Dora.
Okay. So final question from Bruce. What is being done to improve accuracy of cancer diagnostic process, to decrease exposure to ineffective therapies, to make treatments more effective?
It's a great question. One of the things being done for diagnostics, and if you look at cancer diagnostics, what we've always done is used the latest technology we have. Sorry, I hear background. What's conversation? Oh, sorry. We've used the technology at hand to apply to cancer diagnostics. So for many years, it was what we call an H&E slide of a pathologist staining a slide and looking at what the cells look like, or what we call morphology. Then we started getting into the ability to use antibodies that we can label with a marker and look at those under the microscope and start looking at surface proteins on cancer cells. And we learned that different cancers from different organs have different proteins that they express. So with these antibodies, you can distinguish if something's a colon cancer from a pancreas cancer from a lung cancer. Over the last several years, what we've been able to do is look at chromosomes and actually look at the molecular landscape of a cancer, and now even look at RNA and start looking at RNA expression. And we understand that there's different patterns of gene mutations and RNA expression in what we call gene methylation patterns that are unique to different tissue of origin. Because when you look at a cancer cell under the microscope, a perfect example is something called adenocarcinoma. Adenocarcinoma just means a cancer cell that arose from a glandular tissue. But there's glandular tissue in the colon, there's glandular tissue in the stomach, there's glandular tissue in the breast. And all of these have adenocarcinoma as part of it. So how can you distinguish one from the other? And it turns out they all have very specific methylation patterns, as well as those surface proteins I mentioned. The RNA patterns of expression have been very helpful. And we've actually, at one of the companies I'm working with, have developed a way for what we call tumor of undetermined origin. Sometimes the cancer is so undifferentiated, you can't even tell where it originated. And looking at the RNA patterns and patterns we have from other tumors, we can distinguish with high likelihood, with 90+% likelihood, where that cancer came from. So that's a diagnostic issue. The other part of the question has more to do with specific therapeutics, and that's really where we're at now. Cancer therapy has evolved where we take a population of patients and consider them homogeneous. So all lung cancer patients, all breast cancer patients, and we treat them the same way. And we see that these treatments work in 50% of patients and don't work in 50%. And in some diseases, that's fantastic. That's a great breakthrough. In other diseases, it's kind of disappointing. And you can focus on the 50% success rate, or you could say, okay, what's different about the 50% who didn't respond to that treatment, who are getting a treatment that could be toxic? And can we start personalizing this? And this is this whole era of what we call personalized precision molecular oncology right now. And the company I work for, that's basically what we do. And even if we take immunotherapy, the last big Nobel Prize for immunotherapy in medicine, using these drugs you see on television all the time, Keytruda and nivolumab and all these other immune therapy drugs, they're fantastic. And they're helping lung cancer patients live longer. I mean, when I was in medical school, a stage 4 lung cancer patient had an average survival of about 3 to 4 months. And maybe 6 months when cisplatin started getting used more for chemotherapy, but that was it. Nowadays, over 25% of patients and some studies even as high as 30 to 40% are making it out 5 years because of targeted therapies, because of immune therapy, and that's with stage 4 disease. So we've really had tremendous improvement. But the question is, what about all the people not responding and what's going on with them? And can we figure out biologically what's driving their non-response to a treatment and avoid that treatment in those patients? And then figure out who's most likely to respond to it so that we can give those patients the treatment. So that's the stage of cancer research at the clinical level, the bedside level that we're at right now. Not at the bench level, but that's what we're doing clinical studies for right now. And that's exactly right on target. The question itself, how can we start personalizing things to understand who's going to respond to a treatment and who's not? And again, all this has to be validated because we have treatment that has evidence of success. We don't want to deny someone a treatment without absolute certainty that that treatment may really help them, because that treatment's already been established at extending survival. And we don't want to not do it based on a theory that a gene might predict that they're not going to respond. So we do need the validation data. But that's sort of the state of exactly the cutting edge of clinical cancer research right
now. Thank you so much, Dr. Grad. Oh, actually, you know what? I'm so sorry. We said that Bruce's was the last question, but false alarm. There was one more. Jay, how could we miss you? We missed your question. Jay asked just before that, how do you think mRNA will change cancer research? That's the last question.
mRNA is another step in that process of understanding what drives a cancer. You know, right now we're looking at the DNA level, and we can understand if there's a gene mutation. Let's take BRCA2. But the question is that gene mutation always expressed at the RNA level, which is the next level? What happens with DNA is you make RNA, and then RNA makes a protein. When you have a mutation in DNA, you get a different expression of RNA, and you get an abnormal protein. And there's actually some cool research going on by some researchers that we fund in Israel that are looking at how an abnormal protein from a gene mutation, the 3D structure of it is folded differently than the normal structure. You saw the researcher who talked about doing research on P53. P53 mutations results in a protein through the mRNA and then through a protein where the 3D structure of the protein is folded differently. And one of the Israeli researchers that we fund is looking at developing peptides that will react with that protein and reshape it back into its normal, non-mutated form. And the hope is that will overcome the cancer phenotype. So mRNA is that next level. We've been able to use that technology to get to the DNA. We can start understanding mRNA. The problem with mRNA is some of it is directly related to cancer drivers. Some of it is what we call epigenetic, meaning that it's not directly related to driving the cancer, but it has to do with the regulators of the genes that drive the cancer. And so, like anything, once you open Pandora's box and go down the rabbit hole, it's an upside down funnel that just keeps getting wider.
Thank you.
Wow.
Thank you,
Dr. Grad, I know that we have all learned so much tonight, both about cancer research, about Israel, about ICRF, and about the incredible work that's being done. And we thank you for joining us tonight. Thank you so much.
Thank you to Anshiemit. Thank you to Jay. And please, if there's any questions, we're always available at ICRF. So reach out at any time. And everyone have a good Hanukkah and stay safe and healthy, please.
You too. You too.
Thank you so.
Thanks so much, Gary. Thank you, Jay.
Thanks, Jay.
Thank you, Al.